Welcome to Module 1 for the Introduction to Medical Laboratory Technology course. This first presentation will provide an overview of the clinical laboratory departments. This is very important. For each module or unit of instruction, I will provide you with a set of objectives for that module. These are very important in that this is where I get your exam questions. You will be provided with a lot of information, and you as a student are wondering, oh my gosh, what do I need to study for the exam? So what I want you to do for every module is download the objectives. It's in a Word document. Fill in the information that's required of each objective and save it. When it comes time for your exam and when it comes time for the final, all you have to do is pull out your objectives and study all that information. So the objectives for this particular module is that we're going to cover each laboratory department. And what you need to do for exam 1 is provide me with the function of each of the laboratory departments and list tests that are performed in each department. The only tests that you're going to be responsible for are the ones present on this presentation. Clinical laboratories will be found in a variety of settings, and this may include hospitals, large clinics where there's multiple physicians, or perhaps there's a specialty practice, or as a standalone reference laboratory. And these laboratories tend to provide tests that are not cost effective to be provided in-house, but because they're a reference lab and there are so many tests ordered from around the United States that they are able to make money off of those. The number of departments present will be dependent upon the size of the facility and the population served. For example, in an obstetrics gynecology practice, the on-site laboratory testing may include urinalysis, hematology, and some serology. In a hospital, all sections of the laboratory are needed because we have multiple patients with multiple diagnoses who need different types of testing to monitor or diagnose their condition. If a facility has all sections of the laboratory, they will have 7 major departments, and I want you to memorize these: hematology, coagulation, urinalysis, clinical chemistry, microbiology, immunohematology, and serology. These departments may be standalone and separate, or one or more departments may be in one area. For example, hematology, coagulation, and urinalysis are usually housed together, whereas microbiology, due to the unique nature of testing and the requirements, are usually always in a separate area. So here's the answer for your first objective, hematology. Hematology is the study of the nature, the function, and diseases of the blood and blood-forming organs. The most common test performed is the complete blood count, or CBC, which includes a red count, a white count, hemoglobin, hematocrit, and a test called a differential, where we microscopically examine a drop of blood and we count 100 white blood cells and we note the percentage of each type present. And when we get to the section on hematology, I will go into this further. The results of these tests can determine whether or not a patient is anemic, they don't have enough red cells, whether they have a viral or bacterial infection, and this will be noted with the type of white cells that are present, or if they have leukemia, which is a type of cancer of the blood, or many other types of blood disorders. White blood cells are circulating in your bloodstream at all times. There are 5 types. There's the monocytes, and this is a type of macrophage. If you ever seen an amoeba, they move by amoeboid action, and when they find something that is not self or it's a piece of trash, they will engulf it and destroy it. Eosinophils, they're increased in allergies and parasitic infections, so if you have worms, you'll see an increase in the eosinophils, but I primarily associate them with allergies. They have these large granules that will release histamines, which is what promotes the coughing, the sneezing, the watery eyes. So what do you take for that? You take an antihistamine. Basophils are rarely seen in the peripheral blood. They also release histamine that can cause allergy symptoms. They're primarily found in the tissues. Lymphocytes, you'll see these increased in viral infections. Neutrophils, you'll see these increased in bacterial infections. So by enumerating the percentage of each type of white cell present, doctors can determine whether this is a bacterial infection, which will need antibiotics, versus a viral infection, which sometimes we can provide antivirals to patients, but for the most part, viruses have to go through their cycle and make you miserable. For example, a cold. Hematology analyzers. These are the machines that get the job done, and the size of the piece of equipment will depend upon the volume of testing performed. For example, in a reference laboratory that performs thousands of tests a day, they will need larger analyzers which can rapidly analyze large number of samples in a short period of time. And that is what is pictured on the bottom right-hand side of the slide. Smaller laboratories with low volume testing will have smaller instruments which may take longer to produce results, but they can sit on a countertop and do the job for this, for example, a physician office laboratory. The coagulation department performs blood clotting tests used for diagnosing problems with hemostasis, patients who have a difficult time with blood clotting due to a variety of factors. They may be performed for patients who are on anticoagulant therapy, also known as blood thinners. I hate that term. It doesn't thin your blood, it prevents it from clotting. And if a patient is on too much, they can spontaneously bleed to death if they're not on enough. They can throw a clot, which may go to their brain, causing a stroke. Patients who have hereditary bleeding disorders, such as hemophilia, or patients who have excessive bleeding due to some type of trauma. They were in an automobile accident and they have crushed organs and they're bleeding internally. The most commonly performed tests are the prothrombin time, the partial thromboplastin time, fibrinogen, and D-dimer. Here are some examples of coagulation instruments. Just like hematology, the size of the instrument will depend upon the volume of testing performed. Large analyzers for high volume testing, small tabletop models for low volume testing. And I need to interject here that it's the price. These large instruments cost hundreds of thousands of dollars versus a smaller instrument that'll be maybe $30,000 to $50,000. The next department I'd like to discuss is urinalysis and body fluids. In this department, we perform chemical and microscopic examination of urine, as well as other bodily fluids. After blood, urine is the most frequently analyzed body fluid. The most common test performed is the chemical analysis of urine, where you can get a mini physical. It has a dipstick with pads that are impregnated with different chemicals. The dipstick is immersed briefly in the urine according to the manufacturer's instructions, and then the pads are observed for color changes at the appropriate time. And it's very crucial that these pads be interpreted at the time that is stated in the manufacturer's instructions. Urines which give positive results for certain analytes are centrifuged. They're spun around at a high rate of speed, so the formed elements will go to the bottom and form in the sediment. And we go ahead and put a drop of the sediment on a slide and observe for the presence of clinical significant microscopic findings. So the picture on the right shows the urine dipstick, which has all the different pads, and you can see them, they're on the left-hand side of the card. And then once you dip it in urine, then at a specific time you read the results. So leukocytes, WBCs, which is the top pad, indicate a urinary tract infection or UTI. Urobilinogen detects problems with your liver. pH gives an idea of the acid-base balance in your body. Specific gravity is how concentrated the urine is. And we like a first morning sample, which is the most concentrated because you haven't peed all night, well, hopefully, and we can get the most accurate results. So the specific gravity is important to determine if we have a quality sample, as well as some other disease states. Bilirubin is an indicator of hepatitis, which is inflammation of the liver, and there's many causes. There are diseases like hepatitis B, but there's sometimes drugs and other situations that can be toxic to the liver. Protein indicates kidney disease. You should never pass protein in your urine. Blood is an indicator of either a UTI or trauma to the kidneys. This guy was in a bar fight and was pummeled in his kidneys. There's a great likelihood that he could spill blood. Otherwise, you should never spill blood in your urine. Ketones are an indication of diabetes or starvation. And glucose is an indicator of diabetes. You should never spill glucose in your urine. When certain positive reactions are obtained on the dipstick, a microscopic examination will be done. So the urine is centrifuged, the drop of the sediment is examined for medically important structures such as crystals, bacteria, parasites, blood, etc. The picture illustrated on this slide show just a few of the multitude of structures which may be present in urine. Just like the other departments, urine instrumentation will vary dependent upon the volume of testing that's performed. Some instruments will perform the chemical examination only. These are usually small tabletop models. And then we have large volume instruments that not only have the ability to perform chemical examination, but it also does the microscopic examination. The microscopic examinations must be reviewed by a medical laboratory technician or scientist. Clinical chemistry is the largest section in the clinical laboratory, and it is an applied form of biochemistry. It performs analysis on body fluids, most commonly serum or plasma. These are the fluid portions of the blood, and whether it's serum or plasma depends upon what type of test tube it's drawn into during a blood draw. And it's used to determine the concentration of specific analytes in the blood. Testing is usually broken down into panels according to body systems. And this is why physiology is really important to your knowledge base, because the physiology is how things are broken down and substances that are produced. And then these analytes are used to monitor and diagnose disease states. The most commonly ordered test in the clinical chemistry department is the basic metabolic panel, BMP, and the complete metabolic panel, the CMP. The difference in the two panels is the basic has fewer tests than the complete. And they provide information about a number of analytes, which gives an overview of each body system. Microbiology is a branch of medical science concerned with the prevention, diagnosis, and treatment of infectious diseases. Microbiologists study the characteristics of pathogens. Pathogen is something that causes disease. And there's 4 types of microorganisms that cause disease. These are bacteria, fungi, parasites, and viruses. The work performed in the department includes a culture. This is where we take a swab that contains the patient's sample and we streak it or plate it onto appropriate growth media. And the media is designed for organisms. For example, for throat cultures, we'll put it on certain different types of culture media. If it's a stool sample, we'll put it on other types of media. So the source of the sample will determine the culture media, because this will promote the growth of these potentially pathogenic microorganisms, and they're observed over a period of several days. Once the organism grows up, we then perform testing to determine the identity of the organism. What is your name? And then once we identify the organism, we will test the organism against drugs known to kill the organism to determine which one is most effective. I think you've heard there are a lot of drug-resistant type microorganisms out there, and this is due to an abuse of antibiotics. And this is why it's really important that you take your antibiotics for the length of time the doctor prescribes it. Yes, you're going to feel better within 3 to 5 days, but if you don't take it, there's enough organisms left. There's a lower level of the antibiotic that the organism learns to live in the presence of the antibiotic, and that antibiotic then becomes useless. The results of these tests are then reported out to the physician. Molecular techniques and mass spectrometry are dramatically changing testing in this department. It's really shortening the amount of time it takes from 3 days to a matter of hours to get a name for the organism. So as I stated before, the microbiology department is broken down into subdepartments. Bacteriology is the study of bacteria through culture, biochemical testing, and sensitivity testing. Sensitivity is what drugs are they sensitive to. Virology, not many labs do virology due to the unique nature of the testing that's required, is the culture and study of viruses. Parasitology is the identification of parasites from a wide variety of samples, primarily feces. And yes, we have a lot of worms and other types of parasites in the country, primarily in the South, but also due to international travel, you may see parasites where you live. Mycology is a study of fungi, and these are the most beautiful microorganisms I've ever seen. So parasitology and mycology are also no longer routinely done in clinical labs due to the special techniques and the skills that are needed to maintain competence. Due to the complexity of testing involved in low volume, most clinical laboratories will not perform virology, parasitology, or mycology. These are sent to reference labs. So these are pictures that illustrate what we were talking about in bacteriology, the study of bacteria. The first slide shows a culture, and this is where we streak out the sample in different quadrants, so we end up with individual colonies that can be tested. On the bottom side is sensitivity. Once we isolate the pathogen, we make a solution, and we pour it onto an agar plate that has and then we set antibiotic disc on top. If there is no growth around the disc, that means the organism is not killed by that particular antibiotic. And then if you look, you'll see zones, clear zones, which indicate that this bacteria is sensitive to the antibiotic. So we measure the size of these zones and then report out to the doctor which drugs are the best to use for this particular pathogen. Many viral infections such as flu, hepatitis, and HIV can be diagnosed through traditional laboratory testing methods. And now we have COVID-19, and so we have rapid tests that you probably may have received some free ones at home. They're very easy to use and can diagnose a patient very quickly. Molecular testing plays a larger role in other types of viral infections. A sample is collected using very, very special swabs and a fluid to preserve and keep those viruses alive. In picture 2, the viral cultures are grown in a cell type that the virus being tested for is able to infect, and that's what viruses do. They infect cells, and they use that cell's DNA or RNA to replicate within the cell. Once these are replicated, then an electron microscope is used to identify the viruses. This is used for cytomegalovirus, which can cause a deadly infection in infants, varicella zoster, which causes chickenpox and shingles, and rhinovirus, which causes the cold. Parasitology is one of my most favorite areas. I taught this for years, and it's just fascinating. Parasites are present in the United States and are transmitted through a variety of mechanisms: food, water, fecal contamination, vectors such as ticks and fleas. Worms and amoeba are the most common, and they usually cause infections in the intestines. So you can look at the hookworm. Oh my gosh, can you imagine? They use those hooks to hook into your intestinal tract. We have flukes, tapeworms, which can grow 10 feet or more long, nematodes, another type of worm, and then the protozoa, which are amoeba. Mycology, so the study of fungi, and they can cause a variety of infections in various parts of the human body. In the mouth, there's a condition or a disease called thrush that's caused by Candida. Athlete's foot, which is caused by Tinea pedis. Skin, you can get ringworm, and this is caused by Trichophodon. And then the lungs, Aspergillus. Fungal infections of the skin are usually easily recognized, but sometimes cultures are needed, especially for internal infections. Cultures are plated on special media and observed for growth, and this can take days to weeks because they're very, very slow growing. And once they're grown, they are observed microscopically for identification. Fungi are uniquely beautiful, and Dr. Fungus is a really cool website. Once you get into the MLT program and you're studying about the different types of fungi, I would strongly encourage you to visit this site. Immunohematology. This is more commonly known as blood banking or transfusion medicine, and this is the study of antigen and antibody reactions to identify blood groups, determine compatibility of blood and blood components, also known as the cross-match, and determines the cause of hemolytic disease of the fetus and newborn. This is a condition where mother makes antibodies directed against an antigen on a fetal red cell that was inherited from dad. And this can cause severe and sometimes even deadly consequences for the fetus. The transfusion service selects and prepares additional blood components for transfusions, such as fresh frozen plasma, cryoprecipitate, and platelets. We do not normally transfuse whole blood because if a patient is anemic, all they need is red cells. So by giving them too much volume, by including the plasma, could cause additional problems for the patient. Antigens are structures that are present on the surface of red blood cells, and antibodies are substances most commonly known to fight off infection. For example, you've received your COVID-19 vaccine. That stimulates your body to produce anti-COVID-19 antibodies. So when you're infected, these antibodies will attack the virus and kill it. But in blood banking, antibodies are produced against ABO antigens we do not possess, as well as against other blood group antigens an individual is exposed to as a result of transfusion, which we get blood from another individual, it's non-self, or pregnancy, where fetal red cells do escape into the maternal circulation and can cause an immune reaction or response by her. The most important test done on a blood is determining the ABO group. Patients who receive an ABO incompatible blood transfusion will most likely die due to the immediate antigen-antibody reaction, which will occur resulting in the destruction of the transfused red cells and resulting consequences of that destruction. So if you are a group A person, you have anti-B circulating in your plasma. If you receive B blood, then that will potentially kill you. Group O people have anti-A and anti-B, so the only blood group they can receive is Group O. RH or D factor determination is performed as individuals who do not have D will most likely form anti-D, which will impact future transfusions. And so if you are an RH negative woman and you gave birth to an RH positive child, then they will give you a shot of anti-D after birth to destroy those fetal cells that may have leaked into your circulation to prevent you from becoming immunized. There are 4 blood groups, A, B, O, and AB. And AB is the least common. A and O are the most common. Blood typing is performed by adding reagents which will agglutinate, cause cells to clump, if the appropriate antigen is present. No clumping is negative, and it looks smooth and silky, and this indicates the individual does not have the antigen. So if you look at the examples given, these cells were tested with anti-A and anti-B. And based on the agglutination or not, we are able to determine the blood type. So this blood was agglutinated by both typing series. So this indicates that they have both the A and the B antigens, so they're AB. This cell agglutinated by the anti-A but not the anti-B indicates only the presence of the A antigen. And see if you can determine the reason for the other two types of reactions. Compatibility testing, major cross-match, is performed to make sure the donor blood that we're going to transfuse into you, is compatible with the patient. The theory is the same as blood typing, except the patient's serum, the fluid portion of the blood, is added to donor red cells. They're incubated, they're washed, we add a special type of reagent to determine if the patient does have an antibody against an antigen on the donor cells. Clumping would indicate an incompatible donor, and we would not transfuse that blood to that particular patient. The traditional method used in blood bank, which you will learn, is the manual tube method, where reagents and patient samples are added to test tubes, they're mixed, they're centrifuged, then observed for agglutination. Automation is finally making its way into the blood bank. I've been hoping for this. I'm a blood banker since I got out of school many, many years ago. So the advantage of automation, it takes the subjectivity out of an individual looking at reactions, and this can be then interpreted by a machine. So solid phase, the top picture, the endpoint reaction can be read by a machine or by hand. And there's another type of testing known as gel testing, which is the bottom picture, that can either be read by hand or by machine. Serology is testing a patient's sample, usually serum, for the presence or absence of antigens and antibodies. Antibodies are produced against structures which are not self. For example, an important method of preventing disease transmission is through vaccination. A piece of the offending microbe, for example, not the whole organism, but a piece of it, for example, the rubella virus, is injected into your arm. Because it's a piece, it cannot cause disease, but the body recognizes this particular piece, and it's common to all rubella viruses, as foreign, and it mounts an immune response, which results in the formation of antibodies specific for the rubella virus. If the individual is exposed to the virus through somebody having it, their immune system will immediately produce antibodies, which will result in the destruction of the virus before it can set up housekeeping in you and cause an infection in you. And just note, there are vaccine failures. Not all people respond appropriately to certain vaccines, but for the most part, these work. For diagnostic testing kits to determine if an infection is present, these have been developed for the detection of antigens, which is the most specific, or antibodies, which are less specific, as it may take days or weeks to produce detectable amounts. Although the methodologies used are very simple, these are found in all doctors' offices and performed by non-laboratory personnel, the principle, what is reacting with what to give a positive reaction, are crucial in performing the test. Kits have built-in positive and negative controls, which must be interpreted without error. If they don't work, that means the kit has not worked and you can't use those patient results. Many kits require external controls to be run prior to using the kit. So in a lab, one of the first things we do when we use a new kit, pull out those controls, test them, and we have to sacrifice two pieces of the kit, one for our positive control and one for a negative. The steps must be followed exactly. The number of drops, the number of swirls, the timing of each step, if multiple steps are required. The timing is crucial, as false positives or negatives may occur if the timing is off. If you wait If you read it too quickly, the results may give you a false negative. If you read it after the incubation period has expired, you may also get false results. So these must be followed exactly. Serology tests may be spread out by department. Strep testing is done in micro, D-dimer testing in coagulation. So here are some examples. Some tests are so simple, such as the pregnancy test, that these can be purchased by patients at a store. And the pregnancy test used in the lab is more sophisticated. But here you see a C line, which is the control. It must be positive. And this is the test line. This is the patient, which may be positive or negative. Other tests, such as strep and flu, are too complicated to be offered for purchase. Several steps with extraction and testing require specific training. So you can see there's step 1, step 2 with different reagents, step 3 and step 4. Because of the complexity, it is not advised to offer these for purchasing by a patient. In serology, molecular testing is replacing some traditional testing. For example, viral identification as well as viral loads, the number of viral particles present, are the method of choice for HIV and hepatitis C. The viral loads, especially because medications are used with what the ultimate goal being to reduce the number of HIV particles in a patient to where they can live perhaps a full life. Immunochemistry is a high-level analysis of samples using methodologies such as chemiluminescence and fluorometry, which you will learn about in the MLT program, to detect and quantitate substances, either antigen or antibody, that may be present. In summary, the clinical laboratory is a fascinating place to work, and this presentation was designed to give you just a small taste of what is to come. Each of these topics will be covered at length during your journey to becoming a medical laboratory technician. As an MLT student, you'll be trained to acquire the entry-level knowledge and skills to work in each department. As a graduate, you will take a national certification exam to ensure you have achieved entry-level knowledge and skills to practice.